Hi, I am Uwe Hilgert and I'm joining you today from Tucson, Arizona. I am a professor in the Bio5 Institute at the University of Arizona, and my research interests are in microbiology, genetics, bioinformatics, and in biology education. I also teach, and I used to work for many years in Cold Spring Harbor with the great team in the DNA Learning Center. Welcome, and thank you for joining me for a bioinformatics laboratory to identify the cause of sickle cell anemia. You're welcome to follow along, ask questions, and note your responses to the question that I will be asking in a worksheet that you can download, and some of the tools will require that you have the Flash plugin. Bioinformatics enables us to understand how living organisms store and process information in DNA and proteins. In turn, understanding how life handles information will advance our information technology, not only for biology, but also for finance, insurance, and for anything where we need to handle information. DNA is a language that uses 4 letters, just as our language uses some 23 letters, and computer and Morse code use 2 letters each. DNA, therefore, is like a hard drive. It stores information, but it doesn't do anything. Proteins are live stewards. A few things that you probably heard already about in molecular biology, that a gene is a segment on the DNA that encodes a protein. Genes are transcribed into RNA, and RNA is translated into proteins. And that's the central dogma of molecular biology, whereby a 4-letter language DNA is transcribed into a 4-letter language RNA that's then translated into a 20-letter language, the amino acid language of proteins. Now, how is this being done? Basically by means of using 3 triplets in the RNA to place 1 amino acid. And if you go and do all the combination with 4 letters, organize those in triplets, you find that you actually get 64 different possibilities, and therefore be able to place the 20 amino acids comfortably. You even have the opportunity to have several triplets place the same amino acid. That's called the redundancy of the genetic code. Now, 61 triplets place amino acids, 3 don't. 3 terminate their translation at the ribosome, and those stop codons are TAA, TAG, and TGA, whereby in the RNA it would be UAA, UAG, and UGA. One more codon I want to point out to you, it's the ATG. It's a triplet that places methionine, but it's also called this triplet here the start codon because every protein starts out with methionine. Therefore, every coding sequence in a gene starts out with ATG. So I want to go with you now and listen to a person suffering from sickle cell anemia. This video is on the DNLC's Your Genes, Your Health website.
The pain that I'm having right now is crisis pain in both my arms and my shoulders. And I also have chronic back pain from the sickle cell, from damage that that's been done by the disease. And it's really hard to describe the pain. It's not a sharp pain. It's not dull. By no means is it dull, or you can't describe it as an ache. And the only way I could describe it is that it's very hard. It's hard pain, and it's continuous. It's constant. There's no break in it. Like when you have a throb and you have that split second of, where there's no pain and then the other, it just keeps hitting you. It's constant all the time.
So sickle cell is a devastating disease. Let's go to the worksheet now. The first page in our worksheet holds all the information that is at the DNALC Live website about this module. And I would like to point out one bullet here in the resources, and that is the New York Times Weekly had a feature in January 2020 on sickle cell cures. So biomedical researchers are working under high pressure in terms of getting cures for sickle cell anemia. Now, here are a few questions that we talked about already in the first PowerPoint. And here's one extra credit question I would like to point out to you. As to like how many chromosomes do you have to gather at once in order to represent the human genome one time across? So Let's go straight to this, to the foundations of sickle cell anemia, and here specifically the fact that sickle cell anemia plays out in the entire process of oxygen transport in our bodies. Now, what kind of a molecule transports oxygen? It's hemoglobin. It's a protein. Because proteins are the doers in our cells. It's not DNA, it's not RNA, but hemoglobin in and for itself can't transport oxygen. Hemoglobin provides a niche in which a organic cyclic molecule can insert itself almost like a disc. That molecule is called the porphyrin ring. And this molecule, this ring, holds the iron. So hemoglobin provides a niche into which the ring can insert. The ring holds the iron, and then hemoglobin is around that iron to actually prod it to release oxygen. So the iron is actually the atom that binds the oxygen, and it's no wonder. If you have a bicycle that has gotten wet and maybe some salty from some salt that was used on slippery roads, things like that, then you know that iron binds oxygen, and pretty much irreversibly as far as our experience is concerned. As far as the oxygen transport in our body is concerned, this has to be a reversible process. Iron is binding oxygen in the lungs and supposed to release it in our capillaries, in our tissues that need the oxygen. And hemoglobin makes that happen. So besides giving a niche and a home for the porphyrin ring system, and thereby providing the basis for getting the iron into the hemoglobin, hemoglobin is also prodding the iron to release the oxygen. So let's go here to a view of the hemoglobin molecule that we have here at the database that's called the Protein Data Bank, where they used actually different colors for the two different subunits. Hemoglobin consists of 4 subunits, 2 of which are hemoglobin alpha and 2 of which are hemoglobin beta. And in here, I wouldn't know which one is alpha and beta just by the colors, but what I do know is that beta has 147 amino acids and alpha has 142 amino acids. So there's this slight change between the 2 protein types that form a whole hemoglobin, but it's certainly not the color as we might see from here. And then here is a depiction of actually this porphyrin ring and the niche that this globin sequence provides for this porphyrin ring. Now let's go further down and look at another animation here. And that depicts actually hemoglobin in action. And what that refers to is that actually with each in the lungs, with each oxygen that hemoglobin binds, so in total hemoglobin binds 4 oxygen, 1 per iron in each of the 4 subunits, right? With each oxygen that's being bound in the lungs, the shape of the protein changes slightly to allow the next oxygen to bind even faster. What that leads to is actually a higher efficiency of the molecule. Now, as the hemoglobin is located in the red blood cells with tens of thousands of other hemoglobin molecules at once, they're all getting saturated with oxygen while they pass through our lungs. And then with our bloodstream, the red blood cells are transported to the locations where they need to release their cargo now. Now, iron wouldn't release its cargo easily, right? Oxygen at least. And that is then being facilitated by the hemoglobin, the amino acids around the iron that prod the iron to release the oxygen. And at the same time, you see this cooperative action here, whereby with each iron that releases its oxygen, with each oxygen release, the conformation changes slightly for the next oxygen to be released even faster. And so this conformational change is again increasing the efficiency of the hemoglobin. Going back to our worksheet, there is a few more questions about the sickle cell anemia itself, right? It's a genetic disorder. So you can't get it by sharing a drink or the same glass or shaking hands with anybody, but you can get it from your parents. Not by means of that they pass it on to you while you are growing up with them as a child and as a young adult and so forth, but because you receiving your parents' genes through their egg and sperm cells. That makes up you then later. And so this is long done before you are even conceived. That's how you get it from your parents, not because they pass on an infection to you. Now, if you want to determine roughly the chances of getting it, we have to actually do a Punnett square here. And for that, this question is actually relevant. I'm sorry, this one here. What's an autosomal recessive disorder? This is a term that relates to a type of genetic disorders whereby let's say you have a black and white situation with the sickle cell hemoglobin, right? You have either the regular beta globin or you have the sickle cell beta globin, right? And the genes for the regular hemoglobin is on an autosome, not on a sex chromosome, right? So on one of the chromosomes 1 through 22. Then it's a recessive disorder. In other words, in order to have this disorder come out, like in Maya, right? In order for the disorder to come out, you have to have one allele from your mom that is the sickle cell allele and the sickle cell allele from your dad as well. So you have to have it twice. Otherwise, that's not the case. If you have a combination of the sickle cell and the regular beta globin, you will have the beta globin being dominant and will always supersede the action and the protein product of the sickle cell gene, okay? You would not be suffering from sickle cell if you had the heterozygous combination. All right, how can you actually examine this a little bit further? With a Punnett square. And let's go briefly into a Punnett square. We're not completing one here, but Punnett squares are tools to actually determine likelihoods that certain allele combinations are being passed on to offspring by parents. And it's also being used then, depending on what these allele combinations evoke, to actually determine the risk for diseases like sickle cell anemia. But what I want to point out to you is this slight difference in nomenclature, but important. As far as biologists are concerned, the genes are called capital letter HBB and capital letter HBS. The proteins are also called that way, so that makes it sometimes a little tricky when you read the literature and they don't clearly say whether it's about genes or proteins, but it's all capital letters. Now, when we come to Punnett squares and risk for disease, then actually things like dominant recessive play an important role. And in that situation, dominant alleles are always written in capital letters and recessive allele in lowercase letters. So keep that in mind when you think about genes, and we will talk about the genes much more in just a moment, and not so much anymore in the context of this module here, talking about recessive or dominant. But if you do Punnett squares, recessive or dominant are super important. Okay, good. Come back to our worksheet. Want to go and move down here and get an impression what we actually talk about. Not sure we've had this today in this module yet, and that is a regular red blood cell that only has hemoglobins that consist of regular alpha and regular beta globin. They can release oxygen without changing shape. When it comes though to red blood cells that have the alpha globin and the sickle cell globin in each of the hemoglobins, then those molecules can pick up oxygen in the lungs without a problem. But as they are releasing their oxygen, the cells start changing their shape and start clogging capillaries and therefore preventing the downstream areas to be supplied with oxygen, right? So that's then what causing the attacks that Maya was talking about. So what I really am after with this exercise is the following question. What can exert such a strong force that it distorts the red blood cells into rigid sickle cell shaped cells that can get stuck in capillaries? And the hypothesis, since it's a genetic disease, is that we can really go from the DNA and work our way up. Way up to what? Well, to the severe health problems of HBS patients, right? Based on an aberration in the DNA sequence that affects the coding sequence and the message, That affects the amino acid sequence of the proteins, which then affects the 3D structure which they fold into, and which is really the precondition for a functional molecule. An amino acid sequence doesn't do a thing until it's folded up in a three-dimensional structure, and in that, like with hemoglobin, like the globin subunits, can form a niche into which the porphyrin ring can insert itself, can prod the iron to let go of the oxygen. That wouldn't happen with the amino acid sequence unless it folds up into the 3D functional structure. So we want to work through these aspects all stepwise, starting with locating the globin genes in the human genome, examining the DNA sequences for the gene, examining the DNA sequences for the coding sequence, examine the amino acid sequences in the protein and determine for each of those what's different. And then how is the protein shape different between HBB and HBS? To locate the genes for the globin proteins in the human genome, we go to the National Center for Biotechnology Information. The National Center for Biotechnology Information website provides an entry portal for anyone who wants to access nucleic acid sequences, publicly available nucleic acid sequences, whether that's professional researchers from around the world or amateur scientists like ourselves. As this is a professional tool, it is humongous in providing a lot of links to get a lot of different types of work done. And we now, on the right-hand side, want to go to popular resource to the genome to actually see where we find the globin genes. And in genome, we want to go to a data viewer that shows us genomic data, the genome data viewer. Clicking on that gives us access to the entry point for genome data viewer. And you see this beautiful graphic here on the left-hand side that depicts phylogenetic relationships among organisms. And we want to now search the human genome for HBB. And I click this search button, and then it will give me a list of potential hits, potential matches in its databases. And I'm not going to do anything because I know by default it will go to the very first hit, and that's the one I want to work with. So on the left-hand side, we see an ideogram of the human genome, and I want to get this a little larger here. What this provides us is a view of what it looks like if you get the human genome represented just one time across. You need the 22 autosomes, X, Y, and the mitochondrial DNA. And our hit was found, our match to HBBB was found in chromosome 11. So I go back to this view here. And what I want to do, though, is really make sure that I have full real estate and thereby have the chance to examine our results in detail. Go a little higher up here. Oops, maybe we put it down here. And we're seeing here an ideogram just of chromosome 11, whereby I can use the scroll bar to move back and forth. And our match was identified here on the short arm, close to the beginning of chromosome 11, on the short arm. Now, I want to go and analyze our gene structure down here. Could do this, but it's very confusing with all the tracks that are here that we don't really need. So with that, clicking the small X on the on the right-hand side, I am getting rid of these tracks. And now I see the hemoglobin beta gene represented as a set of boxes and connected by lines, 3 boxes to be correct, and 2 lines. Now it's actually the right time to speak about the structure of the gene. The structure of the gene we want to discuss by going to a different website, and that is DNA Subway, right? Some of you may know it already, and now I'm going to move this up again here. And we're not going into DNA Subway to conduct any analysis. On the lower left-hand corner of DNA Subway, there is a button labeled background. When you click on that, you can actually access a multimedia textbook on everything molecular biology and bioinformatics. We want to look at gene structure. And the first image that comes up under structure is actually the depiction here, a representation of the central dogma of molecular biology, DNA transcribed into RNA, which is translated into protein. We want to go now into this one, and I'm not going to click through all the slides here, just slide 10, where it shows us one model for a fundamental way genes are organized. In this case, we're looking at a gene that contains the coding sequence, all the triplets, in an uninterrupted open reading frame between the start codon and the stop codon, and starting with the start codon, ending at the stop codon. Now, what we have here actually is then basically that this entire region, not only the coding sequence, but also small pieces of sequences that they're not getting translated, untranslated regions that flank the coding sequence, being transcribed into RNA and then translated into protein. And that's the open reading frame gene with start codon, stop codon flanking the coding sequence. The second type of a gene model that I want to discuss here with you, that first one, the open reading frame gene, you find in all living organisms, including bacteria, humans, yeast, plants. It's a very foundational gene model. Here now we look at the different type of a gene in this depiction, whereby actually we also have untranslated region flanking the coding region. The coding region, though, is broken up here into 3 pieces and interrupted by stretches that are called introns. So despite the fact that they are not coding, they're not called untranslated, they're called introns because they interrupt the coding sequence. And the pieces that remain and make it into the messenger RNA are called exons. So how does that work? DNA is in its entirety from the start of transcription here to the end of transcription transcribed into messenger RNA, exons, introns, everything. And then messenger RNA is being processed. And during this processing, the introns get removed and the exons get spliced together so that in the end, you actually have, again, a contiguous sequence between start and termination codon, an open reading frame again, right? That's being translated into protein. So let's bring this back to our view that we've seen here. We see that in this case, the directionality goes from right to left. The arrowheads indicate that to me. That's the direction of transcription as well as translation. And we have boxes, exons, and lines, introns. So we have a spliced gene in the hemoglobin beta gene here, right? And the start codon would be somewhere in this region, and the termination and stop codon would be somewhere in this region. And if we now look at the information that always pops up when I happen to hit one of these exons, we see actually that the entire length of the gene is 1608 nucleotides. But of those 1608 nucleotides, we have untranslated region, these lesser intensely shaded boxes here and here. We have untranslated region. Oh, this is not where I wanted to go. Let's see whether we get this pop-up one more time. And then we have the introns here. And if you connect just the darker shaded boxes, this is a sequence of 440 nucleotides of coding sequence. So the last thing I want to do with you now is to move out and just see what the neighborhood is about here. We are actually zooming out, and I click the zoom out button here about 6 times. I don't bother to try to fix this here. I don't know how I actually evoke that, but it's not as important. We still have our regular hemoglobin gene, our HBB here that we just looked at, but then look at this. We have 1, 2, 3, 4, 5 additional genes around here that start with HB. And as you would have guessed, look at this. This is a hemoglobin subunit delta, a hemoglobin subunit epsilon. Oh, we're not hitting it right. And then here we have 2 hemoglobin gamma type genes, hemoglobin gamma 1 and hemoglobin gamma 2. And then here we have a gene that actually doesn't code a protein, and therefore it's called a pseudogene, hemoglobin beta pseudogene. So these 1, 2, 3, 4, 5 genes here, except the pseudogene, actually encode globin genes and beta globin genes. That's what it is. It's not the alpha globin here. And they are actually relevant at different phases in our development. So HBB is the adult beta globin. And I don't know exactly how this works for the others, which one is for infants, which one is for fetus, but they come into being or they become important at different phases in our life developmental stages. Next, we will identify the differences in the sequences of the HBB and the HBS gene. In order to do so, we will go to the tools at the bioservers, a portal to access different kinds of databases. Again, in this case, we go to sequences. I enter sequence server, And then the DNA sequences are sorted into groups. So I have to click Manage Groups to get to where I need to be. Now you see here classes. So these are all classes that have conducted laboratories and have uploaded their DNA to sequence server. And I will change classes here to public. As soon as this database view has changed, here we are now in public, and I find that there is a group that's called HBB and sickle cell anemia. So that entails the DNA sequence that we're going to work with. And we have here access to these sequences. Here is the coding sequence for HBB. If you open it, you would see that, yes, there's indeed nucleotide sequence in here. And remember, we wanted to actually compare the sequences of the genes for hemoglobin beta and sickle cell hemoglobin. So I'm going to change this to the entire gene. And this one here also, the sickle cell to the entire gene. And now I'm going to compare these two. So this alignment will come up very quickly. It's going to tell us that it shows us 500, but that we have a total of 1,814 base pairs that we could be looking at if we would want to, and we want to. So we're going to change this here from 500 to 1814. And now I am going to scroll down just a tad, so that I redraw this alignment on the bottom so that I can see everything. And I am going to make this just a tad smaller so that we see a little more. And I also will change this beautiful face here to the right-hand side. And so what we're having here now, we can go through this entire alignment and search for the differences. Differences being highlighted in yellow. It looks like we have the big difference at the beginning. And then let's just count. 1, 2, 3, 4 more. 4 differences more, right? Now, let me share something with you. And that is, as we are having different sequences in the database, they may come from different experiments, which means that they're not all necessarily starting at the exact same start point. And that's the case in this situation here, that the first sequence starts a little later than the HBB sequence, a little later than the HBS sequence. The way to normalize this is by asking, get that sequence actually trimmed, so that actually overhang like this at either end would be trimmed off. And so now we're starting the sequences exactly with the same kind of nucleotides here. It's sorted into packages of 25. And we would only have those 4 differences that we counted earlier on. Then we can enter this information into our worksheets. There's 4 differences, and the nature of differences was A into, what's happening here, A into G, and further down a T into a C. Further down was it a T into? Yeah, T into C, T into C. Further to the side, an A into a T. And A was replaced by a T in sickle cell. And then ultimately, let's go all the way down to the last change that we saw around 600-ish, right? There's a G into a C. So there's actually 4 nucleotide changes between the HBB and the HBS gene. Now, what do we predict for the coding sequence? Well, it might as well be the same, right? So let's predict, but if we only look at coding sequence as opposed to the DNA, we will see the same situation. Well, let's do the experiment. Let's look at the coding sequence, because the coding sequence might actually be different. I'm clicking done down here to close this. Could actually be different. How can I make that comparison? When it says down here none, I'm going to bring up the HBB coding sequence and the HBS coding sequence. And now I want to compare these two against each other. So I'm going to uncheck the HBB gene sequences and compare just the coding sequences. Now, if you remember from looking at the NCBI genome viewer earlier, you might actually remember that the coding sequence was 444 nucleotides. And how long was the gene sequence? Yeah, that was some 1,600 and something nucleotides, right? And the difference between those two was that the gene sequence contains untranslated region and exon and intron and exon and intron and exon and untranslated region again, right? However, the coding sequence is just the boxes together, and of those, only the shaded areas. That's the 444, so much shorter. So we have exactly this back here, 444 nucleotides, even if we have set this to show us 1814, if we add that. So 444 nucleotides, let's see how many differences. So we have a T to a C and an A to a G. And then if I scroll all the way down, I will find there is no more difference. So if we were now to go and transpose this result, we will actually see that despite we have predicted 4, we only see 2. And what we see actually is in position number 9, the T to C change, and sorry about that. And in position number 9, 18, 19, 20, in position number 20, there is a change, and that was the A to T. So we only have these 2 differences. How come? Well, most likely this A to G that we saw earlier on in the gene is probably in the 3' untranslated region. And then the G to C, which was much further down, remember roughly 600, which was much further down, is probably in the 2nd intron or in the last untranslated region at the very end of the 3rd box, right, that we saw. So it's hard to say. I don't want to go into detail right now. I want to, in turn, spend the time to make a prediction with you. Here in position 9, we have a nucleotide change, and in position 20, we have a nucleotide change. And if you remember what's the connection between nucleotides and proteins, it's actually the genetic code, right? And that genetic code actually holds for triplets of nucleotides. Oops, I went a little too far. Triplets of nucleotides, giving us actually placing amino acids, right? Triplets of nucleotides. So somehow each nucleotide counts. If we go back to this, we could predict that potentially we have this change here too, control copy, but in a slightly different fashion, control V, because position number 9 would be 1, 2, 3, 4, 5, 6, 7, 8, 9, would be the 3rd amino acid, would change potentially, and I don't know right now what it would change from to, and position number 20 would actually be the 7th amino acid that would change from something into something else, right? And that's true here too, would change from something into another amino acid, right? So that's our prediction. Let's see what we find out when we do the experiment. Going back here to sequence server, and I'm going to close this alignment. Done. I'm going to close this alignment and instead show you how you could actually get your hands on the coding sequence. You would open this here and then get yourself the sequence, select it all, copy it out, and now find a tool that translates this for you. Into what? Into amino acid. And such a tool exists here, also in the websites of the DNA Learning Center, and it's called GeneBoy. And the thing is, it actually needs Adobe Flash to run. It's just like you would assume you spell it, so you could even Google GeneBoy, G-E-N-E-B-O-Y. It takes a moment to load it, but here it is. Let's zoom in a tad, and another tad, and a 3rd tad, and 4, and 5. Cool. So the first thing that you have to know is that GeneBoy, according to your mood, you can really change colors up here. So from a coal, a dark color, I'm going to go to a little bit more mystique green color here. Then, in addition to that functionality, GeneBoy holds for you sequences, nucleotide sequences, and allows you to perform operations on those sequences. What I'm after right now is here into your sequence, can I now paste our nucleotide sequence from the HBB gene, the coding sequence. We save this and transform this sequence into what? Into its amino acids. There's our translate. Now, GeneBoy throws us a curveball here, does it? Why? It gives us actually 3 different translates back. What did it do? Well, it started at first with the first nucleotide and parsed it out into 3. And then according to the genetic code table, it placed each amino acid according to its triplet, right? So the first triplet is ATG, and so it placed the methionine, and so forth, until at the end, there was still in the 444 nucleotides a stop codon, so it places an asterisk for that. Now, what are the other 2 reading frames? Well, how would GeneBoy know where to start, right? So it goes just that extra step to say, well, maybe these guys wanted not to start at the first nucleotide, but say at the second nucleotide, right? So if you start at the second nucleotide and parse this then out in triplets, you get another sequence of amino acids that come from that, because you're not starting with ATG anymore. You're starting with TG and the fourth nucleotide, right? So that sequence, the translate would be in reading frame 2 here, and it looks like so, starts with a W, and then it goes ahead and says, well, what do I know? Maybe these guys wanted to start in the third position. So now, what is the third position? It's G, right? G, and then 3 nucleotides and so forth. Parses it out, places the amino acids, and tells us, well, this is when we go to reading frame 3. Why didn't it go to reading frame 4? Well, reading frame 4 is like ATG, and it starts with the next triplet. So it's actually exactly as this here, the reading frame 1 would be, except the methionine would be out, would be missing, right? If you started from the fourth nucleotide, you would not get a methionine. Now, all proteins start out with a methionine. That's the first. Number 2, they're not all over the place interrupted by stop codons, right? So when you look at the second one here, for as an example, it starts out with a W, so that's not a methionine, and it's interrupted by stop codons all over the place. This one starts out with a G here, and so it can't be 2 and 3. It has to be this sequence here that we want to work with, right? So this is GeneBoy. This is a fantastic tool to do the translate for you, and you can now take this amino acid sequence out and start figuring out, like, do the same thing for the HBS, and then take those 2 sequences and go and compare them and try to figure out what's the difference between the 2 amino acids. I want to do this actually in a different tool, and GeneBoy actually wouldn't anyway allow for that. And that tool is called DNA Subway. DNA Subway bundles bioinformatics and tools into workflows that allow users to actually accomplish certain tasks, like annotating genomic DNA, next generation sequencing, and so forth. And the tool that we are after in order to determine the difference between the 2 amino acid sequence, and that is an alignment tool for amino acids, is housed here in the blue line. I'm going to enter DNA Subway with as a guest, and I start a project in the blue line by clicking the blue square, and it's going to be a protein, and I have to enter the amino acid sequences, and either you generated them with GeneBoy and take them from there, or in my case, I have them canned here in a file. I bring them right back here and paste them in. I give it a title, HBB, HBS, and difference between HBS and HBB amino acid sequence. That's what we want to figure out, right? And I hit that continue button here on the lower right hand corner to start my project. Under sequence view, I can have a quick view. Yep, there's my nucleotide sequences, amino acid sequences are right there. And here's our alignment tool that we want to run. But first, we have to select the data that this alignment tool is going to use, and have to save that selection that we made. And then we run it, and it's now viewable, already done. I'm opening muscle here, and I will make it all screen. And I can zoom in here. And it looks like that in those 144 amino acids, that there's only one change early on. And when I go to sequence, I see that that change is in position number 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. It's an E into a V, an E into a V. So I go back to our worksheet, and it's already in here. There's one change, an E changed into a V. So how can that be? I predicted potentially 2 changes. Why? Because we had 2 changes in the coding sequence, right? Position number 9, a nucleotide was changed, and position number 20. But that only leads to 1 change in the amino acid, and that's in position number 7. A glutamate is replaced by a valine. Why didn't we get a change in position number 3, which would be this nucleotide here, T into C? Well, that's a silent mutation. Why is it a silent mutation? Because if you count again, 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's the 3rd triplet in the 3rd position. That's where we have nucleotide change. And if we go back to the DNA, to the genetic code, we see that in many cases, the third position doesn't make a difference. It doesn't matter what nucleotide is in here. And that is also then the case in our situation here, that whatever that triplet here encodes, it encodes the same amino acid, whether it uses a T or a C, all right? So, and therefore we won't find a change in position 3, we only find one in position 7, and it's right here. The question remaining then is, what connects the strong disease phenotype that we've seen with sickle cell anemia with the amino acid sequence? Well, that's the shape of the protein that it takes due to having to perform a certain function, right? So we still have to look at the shape, because now that we've seen all these differences and we see that the mutation leads to disease, for sure, the shape has to be different, right? We are now in the protein data bank, and we'll use the ID number for the hemoglobin beta protein to examine its shape. So in your worksheet, it tells you it's 1IRD, and the search function here gives me several hits here, and I'm going to click right on the first one. And I also know that assembly 1 refers to the alpha globin in the full hemoglobin, and assembly 2 to the beta globin. So I change to biological assembly 2. I scroll down just a little bit, and I want to view the 3D structure of a beta globin. It's coming up right here. So we see here the amino acid sequence of our HBB, and you may remember that in position number 7, well, here it's position number 6 because this tool skips the first methionine. So in position number 6, we have a glutamate. Now, here you see a three-dimensional structure of the entire amino acid sequence, the protein, whereby here is one end, and I don't even know whether it's the start or the end of it. And here's the other end. And between these two ends, there's 144 amino acids stretching out. And these 144 amino acids are of a type and in a sequence that allows them to form a certain structure. In proteins, the amino acids form pretty much two distinctly different structures. One is what you see here, an alpha helical structure, a helical structure, and the other one, amino acids kind of lined up as if they form a sheet, a plane, right? And that would be called beta sheet. Now, in the hemoglobin gene here, in the globin gene, we only find alpha helices back. So there's a helix, a very long one here, then it's connected through a shorter helix here, then there's a connection here to a different helix. So it's a bunch of helixes that the different segments of the globin protein form. Now, hemoglobin has two functions, really. And the first one is to provide a niche into which this organic cyclical molecule, this ring structure, can insert into. It's not binding chemically, but it finds a home in that niche. And then, apologize, this cyclical structure here, the porphyrin ring system, can actually hold an iron that in turn binds oxygen. So the hemoglobin, on the one hand, provides that niche for the porphyrin ring system to bring the iron into the molecule. On the other hand, though, then the second function of globin is to prod the iron to let go of oxygen again. We talked about rust before. It's not difficult to get iron to accept oxygen, to bind oxygen, but it's very difficult to release it. And so if you wanted an idea about the power and what proteins are actually able to accomplish, then you see here, in this case of hemoglobin, that actually the protein is what prods the iron to release the oxygen as soon as it gets into the red blood cell gets into the capillaries. So this is the discussion of the HBB, but what we really wanted to do is we wanted to actually analyze and align HBB and HBS. And the way to do this is we go to Analyze, Sequence and Structure Alignment. Now our 1IRD, I'm going to enter into this window, our 2HBS, which is actually the ID for GenBank to show us the sickle cell sequence. And then we have to go and tell it that we want to see only the beta form, not an epsilon or anything else. And here we already have the beta, so we're good. and select comparison method down here, and we use FATCAD rigid, and we're good to go. We align those two sequences now, and our result is going to come back in pretty much no time. Despite all the calculations that go into this process, we have a result back really fast, and application loaded. And here's our result back. And it tells us that it gives us the sickle cell hemoglobin in an orange type color, and the beta globin, the HBB protein, in sort of a turquoise color here. Let's move down to the alignment, and let's see whether we can get this a little larger. Yes, we can. So in this case, we don't see the fancy structure that we saw before, which is called actually cartoon. We see only a backbone structure, and that backbone structure is like one amino acid here, one here, one here, one here. At each dent in the lineup, there is an amino acid located. And you see again, there is a start or an end here. There's an end or a start here. And the orangey kind of structure is the HBS, and the turquoise kind of structure is the HBB. So, and then the porphyrin ring system. Now we played, I grabbed this with my mouth and turned this around. You see some of the helical structures back, and you know, as much as I turn this around, it's the same shape. There's no difference in the shape. So we're expecting a difference, right? Because we know there's a difference in the amino acid sequence, which leads to disease. And the connecting element is the shape of the amino acid sequences, the shape of the protein. And there, we don't see a difference. Now, what you need to know when you look at computational tools in general is that there is actually errors possible on the side of the providers. Two very simple errors. The first one is that actually the database doesn't have the sequence that you want to analyze. It has maybe the original sequence, but not the mutated sequence. Now, it may have been set so that it shows anything at all. It may have been set to show the same sequence then twice, the original twice, which would explain why this is perfectly aligned here, but if we look twice at the regular hemoglobin beta, right? The second possibility of an error is if something went wrong on the side of the database, and you know they constantly updated, the programs are being updated and debugged and all this kind, and sometimes errors happen. So that actually, just by error, the request to the database, what they show us in the structure view here, is actually leading to the same situation, and that is we see the same protein twice. Now, got to check that out, right? And the way to check this out is we go down here. It provides us actually the sequences of the molecule that it aligned. And we go here in position number 6, because remember, we're missing the methionine. It's position 6. There is in one of them a valine, and in the other one, indeed, a glutamate. So we are looking at an alignment here of the structures of HBB and HBS, but there is no difference. If we go back to our worksheet, we would have to say, while we expect, we don't know what changes, but the nature of the changes should be significant, right? That's what we expect, because how else could this impactful disease, sickle cell hemoglobin, have such a bad consequence for patients? Instead, we see 0 changes, and so nature of changes, none. Shapes are identical. They are the same shapes. So what are we missing? So we now need to look in a little more detail at the amino acids that are taking position number 7 in the HBB as opposed to the HBS. I have here a lineup of the amino acids, of all 20 amino acids, and we can actually go a little deeper into this, magnify this whole thing a little bit. And what you see here is that just like DNA, has a sugar phosphate backbone, these amino acids are being connected to each other by an amino carboxy chain. right? Here's a carboxy group, here's an amino group that connects to the next carboxy group, that connects to through its amino group, through the next amino acid. That's where the term comes from. Here's the amino group, there's the organic acid group, right? Here's the amino group, there's the organic acid, amino organic acid. So this is the constant part for all amino acids. Down here then, you have what's called the side groups for each amino acid, and that's where they all differ from each other. All 20 differ from each other. Here you see for valine, for example, it has a non-charged side group here. There is no positive, no negative charge. It's very neutral with 3 methyl groups. You have here an amino acid methionine that starts every protein off, and it has actually a sulfur in its structure. And then here's an amino acid with a large ring system as in the side group, and that is phenylalanine. Now, the properties that these amino acids get by means of their side groups, whether they are charged or not, whether they are large or small, whether they are hydrophobic or hydrophilic, or polar or non-polar, those characteristics of the side groups are responsible for what shape a protein falls into, as well as what process, what function it can have. Now, I give you only one example. If you imagine 2 phenylalanines being located next to each other in an amino acid chain, you see these large side groups, the aromatic ring system. Now, that large side group would actually force them to somehow seek distance from each other, so that actually the side groups get the space that they need. And so they might actually, as they are connected through the carboxyamino backbone, they might actually form a little bit of a bend, you know, to get away from each other, to give each other space. And that's just one example how actually the physical characteristics, the large size of the aromatic ring system, might lead to a bend in a protein there. Now, let's look really more in detail into valine and glutamate here. And I have another couple images here. There is glutamic acid, also called glutamate, here on the right-hand side. We have valine on the left-hand side. This is the amino acid in HBB. This is the amino acid in sickle cell beta globin. Now, we see some of the characteristics lined up down here, and I want to zoom in a little bit more so that we can actually look at those. And we find here for glutamate especially, that there is, beside the carboxyl group in the backbone, beside the carboxyl group in the backbone, you find for glutamate another carboxyl group, that's the acid component, so to speak, in the side group, versus valine being totally non-charged here. And we see that when we go to the physical characteristics down here, you see actually that the carboxy group, glutamate, becomes very polar, very charged, and therefore actually easily to interact with water. So glutamate is actually also called, and I'm sad that they didn't include this here in the listing. There's one word missing. That would be hydrophilic. It likes water. It's okay with water. It doesn't have a problem with water. It's like sugar and water, not like oil and water. Valine, on the other hand, is like oil and water. It hates water. It's not only non-charged, it cannot even be moved to have a charge in some ways and interact with water. Therefore, it has to get away from water, like oil and water, trying to separate from each other, right? So valine is hydrophobic, and glutamate is hydrophilic. Now, what does that have to do with anything? Well, to tell you the truth, everything. Because we're coming back to an image that we've seen before, and this is here the animation of the entire hemoglobin that shows the cooperativity that occurs when an oxygen is released or an oxygen picked up by the iron in each of the globins. Remember, there's 4 globins in here, and in case of a sickle cell molecule, there is 2 alpha and 2 sickle cell beta. And in the case of the regular hemoglobin, there is 2 alpha and the regular hemoglobin beta. Okay, sorry about that. And so with that conformation change, that explains everything if you keep in mind the differences between glutamate and valine. Now, in the lungs, the hemoglobin picks up oxygen, and the entire shape shifting causes the hemoglobin for each of the individual globins, position number 7, to point the side group into the protein, to point the side group into the inner of the protein. When the hemoglobin then is being transported by means of the red blood cell through the vascular system into capillaries, and the oxygen gets released, the shape shifts again in a way that with that shape shift, the position number 7 points outwards. And here, where this is no problem for HBB, because remember, in position number 7, we have a glutamate. That glutamate is hydrophilic, so when the position is turned away from the inside of the protein and to the outside of the protein, where you have the cytosol, the inside of the red blood cell with all its large water content, glutamate, not an issue, right? It's hydrophilic, it does fine with water. Not so valin. As long as the side group, right here, let's call this the cup here of the container symbolizes the side group. As long as the side group points to the inside of the protein, not an issue. There's not a whole lot of free water in the inside of the protein. But as soon as in the capillary the oxygen is being released from the iron, the shape shifting happens, and the position number 7 is forced to point into the cytosol. It is now, as a hydrophobic side group, it is forced, the valin, to endure there is water. And thermodynamically, it's a very hard situation to have water, but then a molecule that can't be moved to be polar in any way. Now, if it could, I'm sure valin would love to turn back inside, but it can't. There is the whole 144 amino acid of the sickle cell globin is forcing its way onto the valin to keep that way, to point the hydrophobic side group now, the mutated side group that's now hydrophobic, into the watery environment. And the only way, because remember, there's not only one hemoglobin in the red blood cell, there's tens of thousands of hemoglobins in there. So tens of thousands of valins pointing into the inside as the oxygen has been released. And that's where neighboring hemoglobins, right, that are close to each other, find one way to make this actually in a thermodynamically much more tolerable situation. Almost like many oil molecules find each other to build a surface film on water, right? And that is if the valin from another hemoglobin molecule can actually attach to the valin from this hemoglobin molecule, and thereby build a environment that excludes water from the connecting zone. They don't build a chemical bond or anything like that, but it's almost, if allow me that, it's not really like it, because they don't attract each other. But if this is two different magnets, they would attract each other. And by means of connecting this way, associating with each other, they exclude water and thereby generate a thermodynamically and energetically much more tolerable situation. And that happens not only between the sickle cell globin from one hemoglobin and the sickle cell globin from another hemoglobin, but this hemoglobin has another sickle cell hemoglobin that has a valin out there that connects with another hemoglobin molecule, right? And so they're all connecting up all the hemoglobins, building large fibers, and in doing so, actually distorting the red blood cell into that sickle shape. Congratulations. You stuck it out with me. We actually right now solved that puzzle, how a mutation in a gene that leads to a change in an amino acid does not change the shape of the protein, but changes how the protein interacts with its environment. And in that interaction, actually finds a solution energetically whereby it connects with neighboring hemoglobins to build very rigid fibers that distort the shape of the red blood cell to become sickle-shaped. Thank you again. And before you leave, let's watch this video together.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.